Noon. Welcome, everyone. Welcome. Hopefully you were able to participate yesterday in part one. So if you were, welcome back. If not, no worries. Today can still be incredibly valuable for you. This is part two of our two-part series of TEAS prep focused on macromolecules and cells. So yesterday we did content and today we are focused on practice test. So we are designing these questions to be very similar to the TEAS to give you a sense of the TEAS. This webinar will have your responses timed just like you would be taking the TEAS exam. But the difference is we will go over the answers and that way you can see where your strengths are and the areas that you need improvement. And if you were here yesterday, it can also give you a sense for what you retained from yesterday. So welcome and welcome back. I do know that there were some delays yesterday in Facebook, so if you're experiencing any form of a delay, don't panic. It will be there momentarily. There were a few little glitches yesterday, but I think we're doing really great today. I've kind of done a little background before we got started, and so far, so good. But before we dive in, I always like to share with you the many wonderful things that we have here for you in order to ensure that you're as possible. And so I off with just kind of giving you a sense for the amazing resources that we have here uh, for you to help you. Time is limited, and we always want to put our best foot forward with our resources in order to get the most out of them. So yesterday I went over in great detail our question bank here. Our question bank has over 2,000 questions with rationales, and I went over that yesterday. So what I would like to go over today, my next favorite thing of resources on our list are the on-demand videos. These on-demand videos are amazing. So not only is the content, of course, amazing and aligned with what you need to know for TEAS, but I talked about time, how important time is to all of us. And time is so essential that we have this set up to where you can go to your specific topic that you're struggling with. So let's say you need to have a refresher on the cancer connection between the cell cycle and cancer. You could go right to that spot in the on-demand video and spend 29 seconds investigating that. If you needed a refresher on the levels of biological organization, you could go right to that segment in the video and spend 3 minutes getting help in that one particular area. And so I just find that amazing. The whole video is 39 minutes long, and maybe you want to watch the whole thing. But if you didn't, you do have that ability to go to the specific topics. And I think that that is one of the biggest strengths of this particular product. Now, I also wanted to show you, along with these on-demand videos, you get access to cheat sheets and the notes. So check out this cheat sheet. This thing is a thing of beauty. It has all the visuals. Actually, we used this visual yesterday. It has all the visuals from atom to organism, the hierarchy from smallest to largest. It focuses on mitosis and meiosis, just like we talked yesterday, the cell parts and their functions. And there are even a couple of practice questions with answers right down there on the bottom. So that's the cheat sheets. The other part that I wanted to show you was the handouts. So I know a lot of students, if they have the ability to print, even if you print in black and white or if you print them small, you can print these before you watch the video, and then you can do some annotating, you can do some labeling, you can write on it. Like in this one, I would start writing my stages of mitosis just to help me remember. So you have so many different ways that you can interact with the on-demand videos beyond just watching them. Remember that the more senses you activate, the more your brain will remember what you were doing. And so that's one little hint of studying smarter, not harder. So keep those in mind. Those are all things that we have available for you here at Archer Review. So we did the West question bank yesterday, the on-demand videos showing you that we have cheat sheets for every single video. We have notes for every single video. We have timestamps for all of them. Also, if you are thinking, oh, I really need some one-on-one help in a particular area, we do offer tutoring. And I will here send you, let you see what our tutors look like. I don't know why I'm jumping around too fast here today. But this is me. I'm Shellie Harshberger. I'm your science person. So anything you need in the science realm, I can take care of for you. That's Tammy. She's can take care of anything English and reading. And then Brandon, of course, has you completely covered with TEAS math. So if you need a tutor, certainly reach out. As you know, because you're here, we have free live webinars. So make sure you're checking those out. I have 4 webinars next week. So if you are getting down to the wire and you're really pushing it, getting close to taking that TEAS, come to my webinars. I have, they all start 3 p.m. Eastern Standard Time. So I have Genetics and Mendel's Laws, June 10th and 11th. So we'll do content on the 10th and we will do the practice test like today on the 11th. And then I have scientific reasoning content on the 12th and then practice test on the 13th. So take advantage of those. That is free education for you. So certainly join and take advantage of that. Also remember that you can rewatch old webinars. So use that QR code to rewatch old webinars. And this also might be a good time to let you know, if you have an iPhone, you can take a screenshot and all these QR codes will be active. If you have an Android, I don't think it's quite that convenient, but if you take that screenshot, then you do still have the addresses. And of course, you can go to our website and find everything there as well. We have 2 different packages then, the SurePass and the SurePass Pro. And those packages allow you to save a little bit of money and get the resources you need. So the SurePass has our QBank, has our on-demand videos, allows you to track your performance, pretty much everything but the 1-hour tutoring. If you do the SurePass Pro, then you'll get that 1-hour tutoring. Okay, so those are all the wonderful things we have available for you. If you missed anything, I do always bring the QR codes back up at the end, so don't fear. Now, as we get started today, just to give you an idea about how this process works, if you are on Zoom, I'm the only one who can see your answers. So you can feel free to put your answers in there, even if you're guessing or not sure, and I'm the only one who's going to see it. If you're on Instagram, if you're on Facebook, if you're on YouTube, everyone can see your answers, but I still strongly encourage you to participate. Everybody's learning, and so there should be no judgment there. Get out there and get those answers in there. And again, the more senses you activate, the more you'll retain it. And talking about all those social media sources, if you have not started following us on social media, on TikTok, on Instagram, on Facebook, I highly, highly, highly encourage it because we put out practice questions each week. We put out little tips and pointers and we go over content for free each week. So definitely follow us on there and take a look at that. Those are some more other free ways to get prepped for the TEAS. And then lastly, if you need to reach out to us for any reason, email us at prenursing@archerreview.com. Okay, so I think we just might be ready. All right, so the other thing too that we will do, because we want to mimic the TEAS as close as possible, is we will have a timer going. So I will just read the question and then I will set the timer. You can read through the answers. You can put your answer choice or choices in the chat box. Then when that timer goes off, we will go over everything. So without further ado, let's get started. So our first category is macromolecules. Remember, that was carbohydrates, lipids, proteins, nucleic acids. All right, our first question. Which of the following are considered monomers of macromolecules? Select all that apply. All right, let's take a look at our answers. Our correct answers are A, C, and D. And I did see quite a few correct answers out there, so very well done. Let's take a look at why that is. So, monomers of macromolecules. So, remember, those were the individual units that then made up the macromolecule. All right. And remember, the individual unit were things like monosaccharides. Remember that mono stands for one, saccharide stands for sugar. So that was the individual monomer of carbohydrates. Now, the reason that B is incorrect, if we just go down the line, is because notice that prefix poly. Poly means many. Okay, so polysaccharides would really be the synonym to macromolecules for carbohydrates because it's many of those saccharide units, those monomers together. So that would not be a correct answer. Now, C is a correct answer. Remember, fatty acids were the monomers for lipids. Okay, so that is a correct answer, just like A is a correct answer. And then amino acids were the monomers for proteins. All right, so well done, folks. Let's take a look at our next one. Which of the following are characteristics of the plasma membrane? Select all that apply. Okay, are we ready for the big reveal? I saw some correct answers out there. Here we go. A, C, and E. Let's take a look. A is composed of a phospholipid bilayer with hydrophilic heads and hydrophobic tails. And that is true because remember that cells are both in a watery environment and the inside of them are very watery. And so these hydrophilic heads, hydro means water, philic, you can think in terms of water loving, they orient themselves towards the outside watery environment of the cell or the environment it's in, and towards the inside watery environment of that cell. Also, remember that it is a bilayer, meaning that it is made of two layers of phospholipids. Okay. All right. I'm going to go through the correct ones this time, and then we'll take a look at the incorrect ones. Our next correct answer was C. It functions as a selectively permeable barrier, regulating the movements of substances in and out of the cell. Remember this idea of selectively permeable? I joked yesterday and said that the cell membrane is kind of like the bouncer of a really fancy nightclub, and he only determines who can enter very selectively. Well, that's how the cell membrane works. And in this idea of regulation of substances in and out of the cell, it maintains homeostasis. That is a huge function of the cell membrane, to be selectively permeable in order to maintain homeostasis. All right, our final one that was correct was E. The arrangement of the phospholipids helps support the plasma membrane's function. Now, one of the big, broad ideas in biology, in anatomy and physiology, really in health in general, is that structure supports function. So pretty much any of the structures that we will talk about, whether it's an anatomy webinar, whether it's a biology webinar, is how does the structure support the function? So anytime you see that, that is certainly always a true situation. Structure supports function. All right, so let's see what happened with the other two. Why are they incorrect? B. The hydrophobic tails face outward while the hydrophilic heads face inward. Well, this idea, remember, hydrophobic means water repelling. They are afraid of water or repelled by water. And so since the cell is in a watery environment, it doesn't make sense that the part of the membrane that is repelled by water would be facing water. Okay. And so that's what makes that incorrect. D, the phospholipid bilayer allows both water-soluble and fat-soluble molecules to pass through freely. No, so basically because of this polar and non-polar components of the cell membrane, it's actually really hard for molecules to just pass through the cell membrane freely. Some do, but they're generally very small molecules and they don't have an excessive charge, and so they can kind of eke through. Most molecules have to go through proteins that are embedded in the membrane that act kind of like tunnels, allowing substances to pass through. Okay. All right, so that makes that incorrect as well. Well, you guys are doing good. I'm appreciating seeing the answers out there. That certainly makes it much more interactive for all of us, so very well done. Let's take a look at our next one. Which of the following statements about enzymes are true? Select all that apply. All right, I see some correct answers. You guys are doing great. Ready for the big reveal? We got a lot of good correct ones in this one. A, C, D, and E. Those are all correct. B was the only one that was not correct. So let's break it down. A, enzymes lower the activation energy required for reactions to occur. That is essentially the definition of an enzyme. That is their entire job, is to lower that activation energy so that the reaction can occur and expend less energy. All right, so we're going to go over all the correct ones first. C, enzymes bind to specific substrates in a lock and key fashion. Absolutely. They are very unique to the substance that they're interacting with in order to decrease that energy required. And remember, we talked yesterday, if the ranges that they work best in change, it changes their shape and then they can no longer work. And that leads us to the next correct one, D. Enzymes activity can be affected by temperature and pH. Yes, there is a specific range of temperature and pH based on the enzyme. And if the temperature in the body goes outside of that range, or the pH in the body goes outside of that range, then that enzyme will denature. It will break down. It will no longer fit like a lock and a key with its substrate. And so that is one of the drawbacks of enzymes, but they're pretty amazing despite that one drawback. And then our last one, enzymes speed up chemical reactions without being used up. And yes, that is one of the ways that makes them so amazing, because they can just go on to catalyze reaction after reaction after reaction without being used up. So what's wrong with B? Well, if we see, B is actually the opposite of E. It says enzymes are consumed during chemical reactions and must be replaced. That is a completely false statement. They can be used over and over and over. So someone is asking a very good question. They noted that fatty acids and glycerol are both monomers of lipid. If one is there, would it be correct if it was the monomer of lipid? So yes, fatty acid is really what we fall back on as the basic monomer of a lipid, but you're right, glycerol is also a monomer, and generally they are connected to one another on a lipid. It just depends on the particular lipid. For example, our phospholipids that we focused on when we focused on the cell membrane did not have a glycerol. It had those fatty acid tails, but a phospho head, a phosphate head. And so that's an example of where the fatty acid was the only part of the monomer. But you are right. There are many, many other lipids where the monomers are fatty acids joined to a glycerol. Hopefully. There we go. Which of the following correctly describes the structure of DNA? Okay, we ready for the big reveal? The correct answer is C. Let's take a look. DNA has a double helix structure with nitrogenous bases forming the rungs. And remember that this word rungs means the steps of the ladder. Okay, so this is our correct answer. Now let's take a look at the other ones. What's wrong with them? A, DNA is a single-stranded molecule composed of amino acids. So there are two things wrong with A. First of all, it's double-stranded, not single-stranded. And it's not composed of amino acids, it's composed of nucleotides. So there are two things wrong with A. Let's take a look at B. The rungs of DNA ladder consist of sugar and phosphate molecules. No, the sides of the DNA ladder consist of sugar and phosphate molecules. D. In DNA, remember that we just use letters when we pair them. And if you think about what the letters look like capitalized, that helps you put the D together, because adenine is a pointy letter and it pairs with thymine, another pointy letter. They have sharp angles to them. Whereas cytosine pairs with guanine. They're both round, curvy letters. Okay. So that's what works for me. If it doesn't work for you, then, come up with something else. But sometimes it's just the simple little things like, what do the letters look like, that help you put that together? Okay, we are on to our next category, which is cell structure and function. What is the primary difference between vacuoles and lysosomes in eukaryotic cells? Here's the big reveal. Our correct answer is A. Let's take a look. Vacuoles store materials while lysosomes break down waste and cellular debris. Now, if you remember, I said that vacuoles are kind of like our garbage bags. They mostly store waste. I guess they can store some other materials, but for the most part, they're just storing waste. So I think of them like garbage bags. Where lysosomes are more like the garbage disposal, because they're breaking things down, much like you put food in your garbage disposal and it grinds it all up. That's more of how a lysosome works versus vacuole. Now let's take a look at our other responses. What's wrong there? Lysosomes store nutrients? No. Lysosomes' whole job is to just really break down that waste, break down cellular debris. While vacuoles are responsible for digestion. No. And then that's just kind of illogical. The digestion is not one of the functions really of any of our organelle. That is a body system job, not just an organelle job. All right, C, vacuoles contain digestive enzymes while lysosomes store water and nutrients. We would say that ultimately, that's the opposite. We could say that lysosomes contain digestive enzymes because they'll break things down. I don't know if I would maybe fully use the word digestive, but it works. Whereas vacuoles are going to store those items. And I would still say they store more garbage than they do nutrients, but that's neither here nor there. All right, D, lysosomes are only found in plant cells while vacuoles are only found in animal cells. No, actually, plant and animal cells have a vacuole, while a plant cell has one large vacuole. We have lots of vacuoles, and lysosomes are not typically found in plants. And so someone's just double checking. Yes, the answer is A. A is our correct answer. All right, here's our next one. What is the primary difference between the nucleus and the nucleolus in a eukaryotic cell? All right, we ready for the big reveal? Correct answer is C. How'd you do? Actually, from what I see out on all of our services, you all deserve a pat on the back, a nice round of applause, because I think everyone got this correct. So nicely done. So C is our correct answer. The nucleus is what's controlling the cellular activities. It's enclosing DNA, while the nucleolus is producing those ribosomes. And remember, I always just help you remember the difference between the two words by focusing on Leo being in the middle of the name. And the structure that has Leo in the middle of the name is in the middle of the nucleus. So I use that as a way to remember that. So let's break down the others. The nucleus produces ribosomes? No. While the nucleolus stores genetic information, those are just transposed, just complete opposite of one another there. B, the nucleolus is responsible for storing DNA while the nucleus assembles ribosomes, essentially saying the same thing as B. And so those would be switched and then they would work. Okay. All right, then D, the nucleolus regulates gene expression while the nucleus synthesizes proteins. Again, if we flip these, they would be correct, but they're not. They're not flipped, so the only correct answer is C. All right, let's take a look at our next one. Which of the following best differentiates the smooth endoplasmic reticulum from the rough endoplasmic reticulum? All right, let's see how we did. Our correct answer is D. Correct answer is D. So let's take a look. having a delayed reaction with my remote today. That's why it's, let's see if we can just get it to go back. Here we go. There we go. We got it. All right, D, the smooth endoplasmic reticulum synthesizes lipids. The rough endoplasmic reticulum synthesizes proteins. Oh, now what's it doing here? All right. That is correct. That is our correct answer. Now, one thing I'd like to point out here, because I think this word can mess people up sometimes. When you see synthesize in biology, think to make. Okay, so you could reread this as the smooth ER makes lipids while the rough ER makes proteins. Okay, so don't let that word synthesis confuse you. It is just a word that gets used in biology a lot, and I think it can be very confusing. All right, let's take a look at our other ones. Smooth ER stores genetic material. Nope, that's for the nucleus. And rough ER detoxifies toxins. No, that's more of a Golgi apparatus type job. All right, B, a smooth ER produces proteins. No, that would be rough. Rough ER produces lipids. Nope, just swap those two. And then C, smooth ER has ribosomes, rough ER does not. Again, just swap those two, flip them, and then they would be correct. Okay. So I have a question talking about the smooth ER detoxifying, also like a lysosome. No, because the smoothie arm might be able to do some detoxification, but not actually focus on breaking down waste. So like if a toxin ends up in the cell, that would be more shuttled to the smoothie R, whereas waste and garbage would be more broken down in the lysosome. Okay, so hopefully that answers that question. All right, let's take a look at our next one. We are on to microorganisms. Remember that we focused on kind of 5 pathogenic microorganisms that you would see on the TEAS. Which of the following correctly differentiates between microscopic animals, protozoans, and fungi? Select all that apply. I think this is a hard one. I think a lot of the select all that applies are hard. But I know people always comment during these webinars that they kind of, stretch your brain. But if you're taking the T's and you have a select all that apply, let's say that there are 3 correct answers. If you choose 2 of the 3, you still get the entire question wrong. There's no partial credit there. So it's certainly worth stretching your brain on them. All right, so I did have another question that it seemed like there were so many answers going out there that I initially missed, and it was a question about the lysosome. Since plant cells don't typically have lysosomes, what acts like a lysosome in a plant cell? And ultimately, plant cells' vacuoles have kind of that job. So the vacuole being one big, large unit in a plant cell would both store the trash, actually store water for the plant when needed, and it would also break things down. So that large vacuole in a plant cell has many more functions than our vacuoles do. All right, so I hopefully that answered that question and maybe gave anybody who needed a couple of minutes more time on this one to get their answers in there. So let's see our big reveal. Our correct answers are A and B. So let's take a look. A, protozoans are single-celled eukaryotic organisms, while microscopic animals are multicellular. That is correct. I said, remember that protozoans are unique because we don't typically have single-celled eukaryotic organisms. Most eukaryotic organisms are made of many, many cells. They're multicellular, so that makes them very unique. Also remember that protozoans are known as protists. That's kind of a synonym for them, so keep that in mind. Okay, her other correct answer was B. Fungi and protozoans are both eukaryotic, yes, but fungi act as decomposers or parasites, whereas protozoans are primarily free-living or parasitic. Yes, remember protozoans are like single-celled organisms that can exist in like lake water, pond water, but they also can act as parasites, like we saw with the Plasmodium species in the mosquito, causing malaria. Okay, so that's an example of that. All right, so what's wrong with C? Helminths, such as tapeworms and roundworms, are protozoans that cause intestinal infections. Well, we've got two things wrong with this. A, roundworm is not a helminth, and they are microscopic animals. That's how they're classified. They are not protozoans. So there's two things that make C incorrect. Now let's take a look at D. Fungi are prokaryotic microorganisms responsible for infections such as athlete's foot and ringworm. Okay, so here's what's wrong with D. Fungi are eukaryotic, not prokaryotic. And fungi fungi is responsible for the other two. This is actually accurate, okay? But they're not prokaryotic. And so that makes that an incorrect response. Okay. All right, we're moving right along here. Let's take a look at our next one. Which of the following correctly describes differences between gram-positive and gram-negative bacteria? Select all that apply. So we're gonna put a pin in this one for just a minute. I want to go back because I actually made a slight error and I want to correct it. In C here for our previous question, it was incorrect of me to mark off roundworms here. That was my bad. I was thinking ringworm, and that's what made me pause even when I started talking about D. So that's my bad. So helminths are things such as tapeworms and roundworms, but they're not protozoans. And then D does cause athlete's foot and ringworm, but they're not prokaryotic. And that was my error. That was my bad. I apologize. I just mentally had a brain moment where I got those two mixed up. All right, so back to our original question now. We are on the gram-positive, gram-negative bacteria question, and let's see our reveal. So this is another one with 4 correct answers, A, B, D, and E. So let's take a look. A, gram-positive bacteria have that thick peptidoglycan cell wall, while gram-negative bacteria have a thin one. Yes. And the way I kind of think of that is because the wall is thick, it holds on to the purple dye like harder than the thin wall. Because the dye, when you put it in the petri dish or when you put it on the bacteria, it is purple. It's not 100% how it actually works, but it's a way to kind of retain that. It's a way to remember that that purple dye is really going to latch on to that thicker cell wall. All right, our next correct answer then was B, gram-negative bacteria stain pink because their thin peptidoglycan layer does not retain that crystal blue violet. Yeah, it doesn't. It actually pulls on to, there's a pink base in the violet. I think they call it like saffron something, and it is more attracted to that pink base. And so that is why it stains pink. Okay. All right. So our other correct answer then was, I'm trying to remember what our other correct answers were on here, A, B, D, and E. So let's skip down to D. Antibiotics are often designed to target structural differences between gram-positive and gram-negative. Yes, that is essentially the whole reason for the classification and the whole reason that at some point in your schooling, you will certainly be dying bacteria, because that is how many antibiotics target. They target gram-negative or gram-positive bacteria based on that cell wall. All right, and then E, gram-positive bacteria stain purple because their thick peptidoglycan layer retains the crystal violet dye. Absolutely. So it's more attracted to the violet in that dye. So then what's wrong with C? Gram-positive bacteria stain pink due to their thick peptidoglycan wall? No, they actually stain pink due to their thin wall. Okay, that's the difference there. And I'm sorry, that's gram-positive. My goodness, I'm sorry. Shame on me. That's accurate, but they don't stain pink. They stain purple. Okay, so that's what's wrong here. All right, so if it said gram-positive bacteria stain purple due to their thick wall, then that would be accurate. Okay, guys, moving right along here. Why are viruses classified as non-cellular entities? All right, our correct answer is B. Let's take a look. They cannot reproduce or carry out metabolism without a host cell. That's our correct answer. So now let's take a look at the others. They have a nucleus but lack organelles. No, remember that I said they're actually like this creepy thing because they don't, some of them don't even have DNA. They have RNA only. Now, many have DNA, but not all of them. And it just isn't a protein coat. There isn't a nuclear membrane. There isn't a nucleus. There aren't any organelles. There isn't even like a cell membrane. It's a protein coat that wraps around their DNA or RNA. All right, let's take a look at C. They contain both DNA and RNA within their capsid, and that capsid is that protein coat that I was talking about. No, they contain one or the other, but they don't contain both, and that's what makes that incorrect. And then D, they have a plasma membrane but lack a cytoplasm. They don't even have a plasma membrane, and they don't have cytoplasm. They have that capsid that surrounds their DNA and RNA. They're very rudimentary, and they're not even, as we see, they're not even really considered a living being. They're considered a non-cellular entity. All right, let's take a look at our next question. Which of the following pathogenic organisms cause malaria and is transmitted by mosquitoes? Okay, so I had a question. Do viruses have cytoplasm? No, they just really have that protein coat wrapped around the outside of DNA or RNA. Cytoplasm, by definition, cyto stands for cell, and because they're not a non-cellular entity, if they have any type of fluid in there, it would be called something other than cytoplasm. And I'm not familiar with if there's a fluid in there, what it would be called. Also, cytoplasm is a site for metabolism and chemical reactions, and viruses don't have the ability to do that. So that's another reason why they wouldn't have cytoplasm. Whether they have a fluid or some sort of something that kind of holds the DNA or RNA in place, I do not know, but I certainly know it would not be called cytoplasm. Okay, so the reveal for our current question. Our correct answer is C. And I think, knock on wood, almost everybody got that correct. So let's do a nice round of applause. Pat yourself on the back. All right, so C is our correct answer. Remember, protozoans are also known as protists. Now, let's take a look at our others. Virus. Now, a virus is not something that would be causing malaria, and it is not something that then is carried by mosquitoes to cause malaria. Bacterium, same thing. Bacterium is not carried by mosquitoes and it doesn't cause malaria. And then D, same thing with fungus. Not carried by mosquitoes and nor does it cause malaria. So I'm sure that there are some things that mosquitoes can carry. Maybe they can carry, some bacteria and viruses. They might even be able to carry fungus, but they're not carrying, but those are not what causes malaria. So when you put those two together, that's what makes C our correct answer. Okay, we are ready for our next category, cellular processes. So we are moving right along here. We try to keep these webinars right around an hour, and I think we're pretty close. I think we're good on our time here. So let's take a look at our next question. Which of the following occur during interphase of the cell cycle? Select all that apply. All right, we ready for these? I think many of you had the correct answers or some of the correct answers. A, B, D. Let's break it down. A, cell growth and normal functions. B, DNA replication. D, preparation for cell division. Now we're going to go back to my other slide for a minute and talk about these. So normal growth and function occurs right here, and that is G1. And remember that interphase is this whole part of the life of the cell right here. That's all interphase. So that's normal functions, cell growth, so on. DNA replication occurs in the S phase. This is where DNA is doubling so that the cell can ultimately divide. And then during this part of interphase G2 is where it's preparing for the division, it's preparing for mitosis. In that case, it's generally checking for any mistakes or mutations that occurred in the DNA replication process. Okay. All right. So let's take a look at our incorrect answer. Our incorrect answer, well, two of them. C was chromosome alignment. So chromosome alignment is in one of the phases of mitosis. And then separation of sister chromatids, another phase of mitosis, but not phases and not events that occur during interphase. Okay. All right, let's take a look at our next question. Which of the following statements about DNA replication are true? Select all that apply. All right, how'd you do? A, C, and D are our correct answers. Now let's take a look at our question and check out why that is. A, DNA helicase unwinds the double helix and breaks the hydrogen bonds between bases. That essentially is our first step, right? That is our very first step of this process. Now, our next correct answer is C. Each original DNA strand serves as a template for a new complementary strand. That is correct. And I believe I have our illustration. Don't I? Yeah, I'm going to pull back our illustration. So that's what we see in our illustration right here. This was our original DNA, and we have one strand that's going to serve as a template over here, and another strand that's going to serve as a template over here. So that means that each new cell is going to have one old strand of DNA and one new strand of DNA. All right, now answer D. Let's talk about answer D. DNA replication produces two identical DNA molecules, each with one old strand and each with one new strand. I just said that. So that makes that a correct answer. So let's take a look at B. What's wrong with it? The entire DNA molecule is copied into RNA during replication. So this is what's wrong with it. There is no RNA used in replication. RNA is only used in protein synthesis. E, the newly formed DNA molecules contain two entirely brand new strands. No, as we see in our illustration here, we have an old strand and a new strand, an old strand and a new strand. Let's take a look at our next one. What is one process during meiosis that introduces genetic diversity? Well, y'all do need a pat on the back on this one. Everyone got our correct answer. Our correct answer is C. Woohoo! Nicely done. So let's take a look at the incorrect ones, and then I've got a visual on my next slide to kind of go over C for us. So cytokinesis. Cytokinesis is the separation of the two new cells finally in that last step before they are fully two identical cells after mitosis. Not meiosis. Well, I guess cytokinesis occurs in meiosis too. Mitosis does not have genetically diverse cells. That is the type of division that makes two identical cells to the original cell. DNA replication is copying DNA exactly so that it can be the exact same DNA in the two new cells. So now crossing over, let's take a look at crossing over on our slide here. So we have this nice little visual on the bottom to really represent this. So this happens during prophase 1 of meiosis, where these homologous chromosomes line up and then they go through this exchange process where they just swap some of their genes so that some of the information is different on each chromosome. And we see the result of that in the next chromosome here. And that ensures that we are not identical to our parents or our siblings. All right, let's take a look at our next one. Why is it essential that human gametes have 23 chromosomes instead of 46? All right, our correct answer is A. Okay, so let's take a look at why that is. What's up with A? That's to maintain the correct chromosome number after fertilization. So let's break down a couple things before we go over the incorrect answers. Let's remember that gametes are the sex cells, the sperm and the egg. Okay, these are the cells that are the end result of meiosis. Okay, and they have 23 chromosomes instead of 46, so that when we have the sperm with 23 chromosomes and it unites with the egg that has 23 chromosomes, we end up with a zygote that has 46 chromosomes in it. Okay, think about what would happen if we had a sperm with 46, an egg with 46, we make a zygote with 92, then that zygote has cells with 92 chromosomes in them. They meet up with another one and so on, and those numbers would just grow exponentially. So we have to have this number of 23 chromosomes so that when they unite, they have the right number to form the zygote. All right. Okay, folks, I think we are on to our very last question of the day. Let's take a look. Which of the following correctly contrast mitosis and meiosis? Select all that apply. How'd you do? Correct answers are A, C, and D. Let's take a look. A, mitosis produces 2 genetically identical cells, while meiosis produces 4 genetically unique cells. Fully correct. Now, one way that I sillily remember this is that mitosis is making my toes. It's making my body cells. It's making everything but my sex cells. And so we need those to be genetically identical. Meiosis starts with this word me, and it makes the cells that made me. You can think of it that way. It makes the cells that require a sperm and egg to unite to make the person, to make the me out of meiosis. Okay. And in that case, we want genetically unique cells. That's very, very important. All right, C, that's our next correct answer. Meiosis involves 2 rounds of cell division, while mitosis involves only 1. Yes, because remember that after the first round of cell division of meiosis, we still have 46 chromosomes and we need 23. So it has to divide again in order to get those 23. Where mitosis does its division, it has the right number of chromosomes, it's identical to the parent cell, it's good to go. All right, our final correct answer on this was D. The daughter cells in mitosis have the same number of chromosomes as the parent cell. Yes, 46 and 46, while meiosis halves the number of chromosomes. Absolutely, 46 to 23. Now, what's wrong with B? Mitosis occurs in germ cells, while meiosis occurs in somatic cells. We just would need to flop those, okay? Meiosis occurs in germ cells. Mitosis occurs in somatic cells. Remember that germ cells is a broad category name for sex cells, not to be mixed up with germs on a surface, but it's the broad category name for sex cells. All right, so B is incorrect. Our last one, E, both mitosis and meiosis produce gametes for sexual reproduction. No, only meiosis produces gametes for sexual reproduction. So that makes E incorrect. All right. Okay, folks, I always like to wrap up by giving you our QR codes again, just in case you missed them at the beginning of our session. If you have iPhone, just take a screenshot and then all those QR codes are active. If you have Android, do that screenshot so you have the addresses. But I don't think Android is as user-friendly on that. But remember, you've got the on-demand videos and question bank. There are free demos on that. So you can get in there and play around with those before you would have to commit to spending any money. Remember, you have free live webinars. So that's something that you don't have to spend money on either. Rewatching old webinars you don't have to spend money on. So we also, when it's time to spend that money, you've got SurePass and SurePass Pro. Remember that those are packages where with SurePass, you get really essentially everything except for 1 hour tutoring. Whereas if you go with a SurePass Pro, you'll get that 1 hour tutoring as well. And then just to put a face with the names, I'm Shellie Harshberger. I'm your science person. I would be your science tutor if you so chose to sign up. Tammy is our English and reading specialist, and then Brandon is our math specialist. And now finally, if I could ask you guys the biggest favor, could you give me some feedback? Tell me how we did today. Tell me how you felt about the webinar, anything that you would like to see going forward. I would just be so appreciative. So I'm going to sign off here, but I'll leave that up for just a little bit. And I do have a question about osmosis and diffusion. Osmosis and diffusion, check out our chemistry webinars. Osmosis and diffusion ends up being considered a chemistry topic under TEAS. And so I do believe that that is covered in our chemistry webinars. All right, guys, I'm going to sign off, but I'll leave this QR code up for a little bit. Thanks for being a great group. And hopefully I'll get to see you guys next week. Remember, I have webinars 4 days in a row, starting on the 10th, 3 p.m. Eastern Standard Time for all of them. You guys take care. All my best. Bye-bye.